All right, good morning. My name is Liesbeth Veenhoff, and I work in the IRIBA Institute, where our aim is to understand what aging is on a molecular level. And the hope is that by understanding what aging is on this molecular level, we may find the common causes of a diverse array of age-related diseases, amongst which obviously very prominently also neurodegenerative diseases. So in my lab, we take it easy. We don't look at human aging, which is unbelievably complex. We look at aging of the simplest model system that we have in the lab, Baker's yeast. And so aging of a yeast cell is defined as the number of divisions that the cell can do before it dies. And so this particular cell here lives to become 16 because it divides 16 times before it dies. Now, if you would look inside the cell and look what are all the changes that occur within its lifespan, so throughout aging, and you align that to molecular changes in human aging, you'll actually see that there is a large overlap. And we have actually tried out with these guys to map out as comprehensively as possible all the changes that occur with aging. What changes first, what comes later, so what may be cause and consequence. So there's many things that we found, but one of the things that I want to share with you here is this finding, that we find that with aging there is a proteomic drift and there is loss of protein complex stoichiometry. So what do we mean with that? In cells, most proteins, the proteins that do the work, don't work alone. They work in complexes. And so if such a complex would be the bike, it is of high importance that the two genes for the wheels and the frames are read and generated a proper amount of mRNAs, so that in fact the cell can make the proper amount of proteins encoding the wheels and the frame, and so that a functional bike can be assembled and no spare parts are left. So what we see with aging is that in fact on the DNA it's quite okay. On the mRNA level things are changing, but not as dramatically as on the protein level, where we see that the stoichiometry between the components that are needed are off. And so what you're left with is dysfunctional machines that are potentially harmful and spare parts that may aggregate. So this is real data. So on the y-axis is the degree of this stoichiometry loss over aging, and it's just to highlight that in the transcriptome in yellow, those changes are indeed on average smaller than in the proteome level in green. And moreover, that there is some complexes that are changing quite a lot in aging and some that are much more stable. And so we've been working on the nuclear pore complex for the longest time in the lab. And we were very surprised to find that this nuclear pore complex changes a lot in aging. So the nuclear pore complex is the main gate to the nucleus in all the eukaryotes. It regulates traffic. And it's composed of multiple components, 500 in total. And you see that some of the components really go down with aging, whereas others are pretty stable. So this is an example of a protein complex that becomes very substoichiometric. Irina has a poster on this topic that you should have a look at, because as you can expect, this should have an impact on the function of the nuclear pore complex. And she's been measuring this, and she can tell you all about it. Now, from a completely unexpected side, namely from the side of people interested in neurodegeneration, which was not our take on it, over the last years, there's a huge amount of data that came out that say the nuclear pore complex is impacted heavily in neurodegenerative diseases. So we got to the nuclear pore complex from the aging angle. Others come to the nuclear pore complex from the neurodegeneration angle. And so this is, for us, the main question for the coming years, is how is the nuclear pore complex exactly impacted in maintaining protein homeostasis, in normal aging, and in neurodegeneration. So if you think of it, it's very logical. The main task of the nuclear pore complex is to regulate transport of transcriptional regulators, transport of mRNAs. It's involved in synthesis of ribosomes. And by all these tasks, it directly defines what proteins are made in the cytosol. So it defines the proteome of a cell. So failure to do all this transport correctly will result in a loss of protein homeostasis. But now if you flip it around, the nuclear pore complex with its 500 components is an incredibly difficult complex to make. And so failure in any of the processes that normally guarantee protein homeostasis will very heavily impact the nuclear pore complex and will result in dysfunctional pores. And so we have here sort of a feed-forward loop of bad things happening, deregulating the proteome. And with that, this is the main question for the coming years, and I decided, I see I have time left, 30 seconds, I decided to focus on the main questions for the coming time, because it's my strong belief that it's not answers that drive science, but it's the questions that we ask. So I invite you all to be very critical about the exact questions that we're asking, because that's what it's all about in science. And with that, I want to thank the current members of the lab, some that have left, and all of our international collaborators. Thank you for your attention.